Viruses, bacteria, and archaea. So are all three of those the same organism? No, viruses are vastly different than bacteria. Viruses, for one, are a magnitude smaller. What does that do to viruses and your chances of getting one? Does it make them go up or does it make it go down? Yeah, how come? Yes, and they're what we call airborne. Most bacteria are so heavy, like if you're talking and spit's coming out of your mouth, bacteria are so heavy they're going to go to the floor. So if you don't lick your shoes or something like that, you're going to be what? Okay, but viruses are so small, just somebody talking, they're airborne. They can travel straight through what? Through the air. Because they're so teeny tiny, it makes them a lot more easy to be spread. Very, very hard to keep viruses from spreading. So we start off talking about viruses. And we knew for a long time viruses were there, but they were so small we couldn't see them. So how did we know they were there if we couldn't see them? Yeah, we could see their effects, right? We could see if one person in the family came down with a sickness, everybody else came down with a sickness. We knew there was something there causing it, but it was so small we couldn't see. When was the first time we started seeing viruses? It was with the invention of something. What did we have to invent to see viruses? The electron microscope. So has a regular microscope been around for a long time? It has. They kind of started out as magnifying glasses, and we kind of called them the primitive microscope, and we've gotten better and better and better. But not until the late 70s, early 80s did we invent an electron microscope. So have we been looking at viruses a long, long time? We haven't. We've only been able to see them for a few years as far as the whole history of things go. Do you think there's viruses out there that we haven't seen yet? Definitely. And we think that a lot of diseases that have unknown causes could be due to what? Viruses that we just haven't seen. So this guy right here was the guy that started it off, Louis Pasteur. He was a Frenchman, and he lived in the 1880s. He was studying rabies. Rabies is caused by a virus. It's very much smaller than a bacteria. Could people in the 1880s see the effects of rabies? Yeah, so how many of you have a pet that you get rabies vaccinated every year? Yeah, I hope everybody who has a pet does that. Because if you actually get rabies, what is your chance of survival? So if your dog contracts rabies, now it's got rabies, what is its chance of like overcoming that virus? None. It will what? It will die. What is your chance of overcoming rabies? It's like a point. The only case that has been documented of somebody surviving rabies happened about 7 or 8 years ago. There was this little girl, she lived in Texas, and she was out in her backyard playing. She was actually being raised by her grandparents, so she was living with her grandparents, and there was this cat that come up into her backyard. She lived in a subdivision. What do little girls like to do with cats? Play with them, right? So the little girl went and started playing with the cat, and the cat was rabid. It was actually a feral cat. It belonged to nobody. Nobody had vaccinated it, and it had contracted rabies. And it bit the little girl, and the little girl run in and tell her grandma, this cat bit me. And what did her grandma say? Quit messing with the cat. And she didn't really pay her any what? Any attention. And the thing about rabies is if you get vaccinated for it, after you've been exposed, you've got about 10 days. Is that a pretty good incubation period? It is, because you can decide, hey, I better go and get vaccinated. And if the grandmother had vaccinated the little girl, she would have not gotten it. But she didn't, because she didn't really think it was that big of a deal. I'm probably that kind of parent too, because like my kids come in like, you're okay. Get you a popsicle, watch you some SpongeBob, you're okay, right? Yeah. So that's kind of like what happened. Well, in 10 days, that virus had become full-blown rabies virus, and she then was showing all the signs and symptoms of having rabies, which is your brain starts to swell, you have an elevated temperature, you pretty much go into a coma, and your brain continues to swell, and then you die. That's pretty much the way it runs its course. So this grandmother took this little girl to the hospital, and basically there's no like miracle drug. We can't give you a shot at that point. They just had to monitor her and try to keep her symptoms from damaging her brain as much as possible. And this was on the nightly news, so like every day I would be watching the news and they would say, the little girl from Texas is still alive. This is the longest anybody who's ever had rabies has lived. The little girl is still alive. Doctors are astounded, but she's still alive. She does have brain swelling, but she's still alive. And then her brain swelling started to go back down, and she actually come to. And I remember the night they were interviewing the little girl on the news. She was about 8 or 9 years old. And when they were interviewing, they were like, you know, hey, we're so glad that you've come back too. And when she was talking like someone who had brain damage. Have you ever known somebody who's had a traumatic brain injury and their speech is slowed? And I was like, ooh, even though she came back, she's not going to be what? The same little girl as she was before she went in. But it was like a medical miracle because nobody else had been documented to actually what? Survive it. Yeah. Now, somebody told me, I have not seen it. I try to stray from this show, but have y'all ever saw that show, Monsters Inside Me? They said they did one on her, and that she was perfectly fine. She had totally overcome it, and she was perfectly fine. I don't know. I didn't see it, but I would like to see it, but I just can't watch that show. It terrifies me too bad. Yeah. So rabies in his time and even in our time is a big thing. If you contract it, you've got 10 days to get that vaccine, or pretty much you are going to what? You're going to die. Yeah. So he was very interested in it, and he was interested in it because people he was seeing were dying from it. He named it a virus. The virus means poison. So even though they couldn't see it, they knew there was some type of poison in these people that was causing them to have these problems. So I do believe on your test you will have a question that says, what is the actual meaning of virus? And you're going to say what? Poison. That's right. So this guy here, Dmitry Ivanovsky, he was a Russian, and he was also a little bit after Pasteur, 1892, and he was studying a virus that affected tobacco plants. Why? Why would he care if the tobacco got a virus? Yeah, and what did he depend on tobacco for? What did a lot of people in the South depend on tobacco for? Money. That was their cash crop. They sold tobacco to pay for everything they had. So if their tobacco looked like this one day, and it looked like this and this the next day, he knew he couldn't sell that tobacco, and he was going to what? Lose money. Everything's always driven by what? The green stuff, right. So he's like, we got to find out what's happening to our tobacco because we cannot keep losing crops because that's how we're going to live. So he figured out the tobacco mosaic disease. They called it tobacco mosaic because it caused the tobacco to have this real weird leaf shape. And that leaf shape made their tobacco unsellable. So they knew whatever was in their fields. Did y'all know plants could get viruses? They can. And that's what tobacco virus looks like. Yeah. So he knew that it might be bacteria, but when he did the test and filtered out all of the bacteria, it still had it. So he knew it had to be something smaller than a bacteria. They knew it had to be there, but we still couldn't what? We couldn't see it. We could not see it. So he filtered out the bacteria, but the extract still caused the disease. And here is the invention of the first electron microscope. Have any of y'all ever saw an electron microscope? I haven't either. And I've worked in several different labs, worked in a hospital lab, worked in up in Huntsville at a research lab. But the thing about electron microscopes is it zooms you so far in. The labs that I worked in were looking at bacteria, and we've already said bacteria is way what? way larger. So we didn't need an electron microscope because everything that we did was bacteria size or bigger. Yeah, but this is what the first ones looked like. It is the electron microscope. We saw viruses for the very first time. They are extremely small. They're the size of a large protein or a macromolecule, about 200 nanometers. Nanometers is down that scale a couple of slots from the size of a bacteria. And here are pictures. So are viruses alive? There are people who say they are, and there are people who say they're not. And that's scientists and all kinds of people. Why would you say they're not? I mean, they make us sick, they multiply, we know they reproduce in a human or in a tobacco or wherever they're in. Why would anybody say they're not alive? Because they don't have enough DNA in them to replicate on their own. If I had a virus and I laid it on that table, it wouldn't matter how deadly it was. If it stayed right there on the table, it would never get any bigger. It would never get any smaller. It would sit there dormant until it got in a host cell. So viruses require your DNA. When they get into your cell, they take over your DNA and they start making copies of themselves. Without your cell, they cannot make a copy of themselves. So most scientists believe that viruses are non-living, even though they have a lot of characteristics of living things once they get inside your cell. If you ever go to the CDC, you can't just go there. Like, you've got to have a reason, like work there or whatever. They have safes and safes full of viruses, deadly viruses, even Ebola and all of the really, really bad ones. They're in containers that are closed. If you go in there, you have to suit up where that virus could not get in your cell. As long as it's in that container, is it safe? It is. Because it cannot make copies unless it's inside of what? Right. Why would we want these viruses in containers? Yeah, and we can study them. So they can protect themselves, but put it in something like a mouse cell and see what happens. And we do that so that if it works with a mouse, maybe we make a vaccine that'll work with a human. We can, yeah. So they're vastly different in shapes. They almost look like, to me, they look like outer space or something. They usually always stain them very bright colors, and they do that by what protein's there. They have many different kinds of weird outer coverings, and that's why you have to take a flu shot every year, because they do what? They mutate. They change its shape just a little, so next year you can get the flu even if you had the flu this year, because it's not the same flu. It's slightly what? Slightly different. Yeah. This right here is an HIV virus. You can see it's got multiple sides. We're going to talk all about that as we go. Yeah. All right, so viruses, living versus non-living. I may have overspoke. I probably should have waited. So viruses have no membrane of their own. They have no ribosomes. They have no cytoplasm. They have no chloroplast or mitochondria. So you guys just finished the cell, I know, because I'm teaching 103 too. Maybe it was a couple of weeks ago. What is a mitochondria and what is it used for? Yeah! When I came through college, we called it the mighty mitochondria, because it gives us your what? Your energy. She said powerhouse of the cell, and that's probably what every one of you heard in high school biology. Powerhouse of the cell. So if the virus has no mitochondria, he is making no what? No energy. How can he do anything? He steals the energy from who? From you, from your cells. That's why he's got to get inside your cells. That's the same thing with ribosomes. Do y'all remember what ribosomes do? They're the site of protein synthesis. Yes, he doesn't bring ribosomes. Whose does he have to use? Yours, right. He has no source of energy. He cannot move or grow on his own. He cannot reproduce without a host cell. The only way a virus can reproduce or make copies is if he is inside one of your cells. Yeah. So virus equals ultimate simplicity. A virus is the tiniest amount of usually RNA or DNA, but not both, that can take over your cells. Everything else he's going to get from who? From you. Yeah. Everything else he is going to get from who? From you. He's like the ultimate light packer. He's only got the bare necessities that's going to allow him to take over your cell and make your cell work for him. So outside of our realm of living things, they're not classified. So do y'all remember prokaryotes? Who falls into the group prokaryotes? Wait. What does prokaryotic mean? No membrane-bound nucleus. Yes. So he don't even fall into that group. He's not even considered living by a lot of people. So viruses equals obligate intracellular parasite. So in your notes, I want you to write, there is no such thing as a good virus. All viruses are parasites. So somebody tell me the definition of parasite. Do y'all know what a parasite is? It lives in or on another cell, causing that cell harm. Parasites always cause what? Harm. All viruses are parasites. But let's talk about these two words before it. Obligate. What does that sound like? Yes. What does obligate sound like? I mean, what does that mean? It sounds like obligate, don't it? If you're obligated, you what? You have to. So obligate means you have to. Intracellular means what? That's right. So a virus is obligated to live where? Inside the cell. And when they do, it causes that cell what? Harm. And that's why we call them an obligate intracellular parasite. Yeah. So one, they must use the host to replicate. They can't make a copy of themselves unless they're inside your cell taking over your DNA. They too are very cell specific. So you cannot get a dog virus. You can't get tobacco mosaic virus. But some of them, the group that they're specific to is really big. So can you and a dog get rabies? You can, because rabies only thing is it has to be a mammal. So could a raccoon get rabies? It could, because it's a mammal. So that group that it's specific to is very, very large. But some are very, very specific. Parvo. Puppies get parvo sometimes, and you can have a puppy with parvo, and a lot of times, especially if you don't get it treated, that puppy's going away. Yeah. You can treat that puppy, you can sleep with it, you can have it. You're not going to get what? You're not going to get it, because that virus only attaches to that specific host cells. And the virus evolves after the cell is what we believe. And that's why she's got a question mark there. So virus evolved after the cell, so it evolved to fit that specific type of cell. So components of a virus. You got to know this for the test. All viruses have what we call a capsid. It's an outer covering, and it's made of protein. So capsids are what? Outer covering, and they're made of what? Yes. You also have an inner core, and it says nucleic acid. So tell me what a nucleic acid is. Yeah, you're getting so much more specific. You are 100% correct, but it's either deoxyribo what? Nucleic acid. What is deoxyribonucleic acid? DNA. Or it's ribonucleic acid. And that's who? Yes. So when we're talking about this inner core, viruses are either DNA viruses or RNA viruses. They generally do not have what? Both. Does your cells have both? Yeah, y'all remember transcription, translation? RNA, messenger RNA, tRNA, yeah, DNA. Yeah, viruses are not like that. So they've all got a capsid that's an outer outer coat covering, and it's a protein. And then inside it, they have an inner core, which is either DNA or RNA. And then they have a few hundred genes, which, you know, when we say several hundred, that sounds pretty big, but you've got millions. So that's very, very simple. And then some, not all, but some have a membrane outside the capsid. And for my test, that is a question. What do all viruses have? You would say what? Capsid and... Y'all quit talking to me. A capsid and a what? Inner core. Yes, all viruses have those two things, every one of them. What do some viruses have on top of that? That's right, a membrane outside the capsid. And the reason I'm making such a big deal about this is a tricky part that viruses have. So sometimes, most of the time, especially in people y'all's age, and I'm going to go ahead and throw myself in the same group with you, healthy, pretty young folks, your body gets invaded by a virus. Before it enters the cell, your immune system goes, hey, invader, and you do what to that virus? You attack it, you eat it up, you stay healthy. You never even knew that you breathed in a virus because you didn't get sick. But what this outer membrane has done is some viruses have become very sneaky. They will come into your cell and take a little piece of your plasma membrane with it and coat themselves with it. So now, when that virus bumps into your regular cell, it thinks it's who? Another cell. So it don't what? It don't attack it. And that gives him more time to get in your cell so that he can take over the DNA, and then he start making thousands and thousands of copies just like that. It's a sneak attack. So this is what it looks like. So this is the adenovirus. It's a DNA virus with a polyhedral capsid. What does poly mean? Many. Polyhedral is many sides. So you can see this is the capsid. All of these blue circles. What did we say the capsid actually was? Protein. And then this one is a DNA virus. What did we say this inside it was called? An inner core. Yes. Could this, or is this always DNA? No. Sometimes it's what? RNA. Here is another example. This right here is called a bacteriophage. Does anybody have any idea what in the world a bacteriophage is? It's a virus that infects bacteria. It's a virus that infects bacteria. So I have a little story here. They tried these right here out as a treatment. So if you have strep throat, that's caused from a bacteria. If we could give you a virus that infected that strep bug, could it kill it? It could. And if that virus didn't affect your cells, would it bother you at all? It wouldn't. So they called that nano treatment. They did it not for strep throat. I just say that one because we've all kind of had strep throat, but they tried it for cancer. So it was similar to that. It was very similar. It was on that same line. Yeah. That was, yeah. And kill the cancer cells too. Interesting. This is like one step down from that. So what they were doing is they were injecting the virus with some medication. That virus was specific to cancer, so it wouldn't stick to any of your normal cells, but it would just go straight to the cancer cells, and it would dump the medicine on them. And so it was supposed to not make you lose your hair, not make you sick, not because it didn't affect any of your normal cells. It only affected the cancer cells. But they found that it worked great, but it only worked for like a couple of weeks because your body is so good at recognizing what? that your body, your immune system, would destroy it before it ever had a chance to get where? To the cells that you wanted it to affect. Yeah, but that's interesting. So, yeah. All right, so here's just a couple of other examples. This is the tobacco mosaic virus. So what are these blue things? Capsid. And what is this? The inner core. This time it's not DNA, but it's what? RNA. And that is the tobacco mosaic virus that he was looking at. And then this right here is the flu virus. So what is this blue stuff? That's the capsid. And then what is this? The inner core. Yeah, which is RNA. And then what is this got? That's right. It's got that big, thick envelope that helps disguise it and protect it until it gets where? Yeah, right, to the host cell. And this is the flu virus. And what happens is this part don't change every year as much as this part changes. So if this part changes, and that's where you hear H1N1. Have y'all heard of that? What was the swine flu? It had H5N1. I can't remember. Do y'all remember swine flu? Y'all were all probably just little kids, and they shut some schools down because so many people had the swine flu. That H and that N are descriptive of these. Sorry, y'all. I'm going to have to get some time. What do all viruses have? Inner core and, that's right. What do some viruses have? That's right. And here they call it an envelope. Yeah. All right. So here is how viruses are. They're kind of like a lock and a key. Can you stick any key into a lock? You cannot. It has to be specific. So viral reproduction equals a lock and a key. The capsid has to adhere with the receptors on the host cell. So if you don't have receptors for that specific capsid to stick to, that virus is not going to what? Not going to infect you. It's got to stick to the cell first. So viral reproduction is like a lock and a key. The capsid adheres with the receptor on the host cell. Very specific viruses are. So here's the little virus, and he says, I'm taking over this cell, and the cell's got his arms up like he's scared. So once inside the host cell, it takes over the metabolic machinery of the cell. Sorry, y'all. What does that mean? Metabolic machinery. What is metabolism? All the chemical reactions that's going on in a cell, and that's what this says. Once a virus gets in, he says, okay, liver cell, you're not going to do what a liver cell does. You're going to start working for who? For the virus. Yes, he takes over it, and he says, okay, liver cell, you're not going to do what you used to do. You're going to start working for me, and now you're going to make more viruses, because that's what viruses do. They make more and more and more and more and more of themselves. So what do you think? You think it takes a long time to make a virus or a short time to make a virus? How come? That is correct. She said short. Why would it take just a short time to make them? Are they real complicated? No, they got a tiny little strip of DNA, and they got this protein, and basically that's all they what? All they are. That's all they are. So, bacteriophages. Me and you are going to look at this whole process, and I'm sure you've got several test questions about this process. We don't talk about viruses infecting human cells. We talk about them infecting bacteria. Why? Because this process is quite a bit less complicated. If you understand how they affect a bacteria, you can understand how they affect a human cell. But you got to see the simple way what? First. So, what is a bacteriophage? A virus that infects a bacteria. Yes. So viruses that parasitize bacteria are studied much. Why? Why would we study these things? Why don't we study the viruses that infect us? Yeah, because if we understand how it works with the bacteria, then can we apply it to a human? We can. We can't just go out there and say, okay, let's infect all these human cells with this virus and see what happens, right? Can we do that with bacteria, though? We can, because we can kill bacteria very easily. We know they're very tiny, so we got them all contained. We can do that. So there are two life cycles that are observed, and you got to understand both of these. The lytic cycle has 5 stages. That's one of the cycles. The lytic cycle is the first one. First is the virus has to do what? Attach. Yes. And then it has to penetrate. That means it has to get into the cell. It has to go in. And then it does what your book calls biosynthesis. Basically, that means it starts using your cell to make more viruses. Synthesize means to make, bio means living, so it's making the parts to make this virus. And then the virus has to mature, so that stage is called maturation, and then release. And we're going to see, I believe, a picture of this on the next slide. Miss Breen has such good PowerPoints with stuff moving, and yeah. So you guys tell me, what's the first step? Attachment. So right here, what is this thing? Yeah, what kind of cell? A bacterial cell. What is this thing? Yeah, that's, what is the capsid part of? That's right. So this is a virus. This virus has to do what to this bacterial cell? Yes, it has to attach, and then it has to put its DNA in. Do you see how this whole part stays out? The only thing that goes into the bacterial cell is the what? The DNA. Now, viruses that infect humans, sometimes they do it this exact same way, and sometimes that whole entire virus does what? Goes in. Right. But once this DNA or RNA gets in, it takes over this thing. What is this? Yeah. It's a chromosome, but we don't call it a chromosome because it's in a bacteria and it forms a loop. We call it a plasmid. So that virus gets into that plasmid and says, hey, bacteria, instead of doing whatever you were doing, you're going to start making what? More viruses. Right. And it does it in parts. Does it make copies of the DNA? It does. Does it make copies of this outside coat? It does. And then them have to get put together, and we call that what? Maturation. And then once they're fully, completely mature, they're going to bust that bacteria cell open, and all of them are going to what? That's right. They're going to go find another bacterial cell, and they're going to infect some more, and this process is just going to keep on going until this bacteria figures out a way to what? That's right. Kill that virus, or don't let it in, so that it doesn't get in, because once it gets in, it's going to take over that DNA, that bacteria's DNA, and it's going to tell it, all you got to do is make virus, on and on and on. That was the one. That was the lytic phase. Now we're going to look at the lysogenic cycle. So the lysogenic cycle is very, very similar. The first steps are the same. So what happens is, and I think we have a picture that shows them together in just a minute, is same thing. Attachment, exactly the same. Penetration, exactly the same. But this time, instead of taking over that DNA altogether, it just incorporates itself into it. And it goes latent. Does anybody know what latent means? Resting. So this bacteria now still doing what the bacteria does, but instead of just having his own DNA, he also has some what? Virus DNA. So if this guy makes a copy, he makes a copy of his own DNA, but he also makes a copy of what? The virus DNA. And he does it again and again and again and again, but then at some point, something in nature triggers this to become active. And now instead of just one cell with virus in it, you're going to have what? A bunch. And they're all going to be activated at once. And so they're all going to start making virus at once. So phage becomes prophage. That means before. So this is prophage where it's just incorporated in. And then it becomes latent. Latent means what again? Resting. It's making copies, but they're not active virus yet. And then it says later they may re-enter the lytic cycle. So here it is together. Up here is the loop for lytic cycle, where, what's the first step? Attachment. Who attaches to who? That's right. Virus attaches to bacteria. Second step is what? Penetration, where what comes in? DNA or RNA. Yep. And it takes over. And then what's step 3? Yes. If we've turned off the bacteria cells and started him making what? That's right. More viruses. And then these viruses have to mature. And now we got mature virus, and they what? They release. They exit. They leave the cell. But in some cases, we still have attachment. We still have penetration. But what happens? That goes latent. If the bacterial DNA incorporates the virus DNA in it, but it doesn't turn into virus yet. It continues on, and it divides, and it divides. This just shows it dividing one time. Is that really how bacteria would do it? Nope. These would divide, and these would divide, and you'd have 1,000 of them. And then all of a sudden they get the message, and all of them go to biosynthesis. So instead of one cell making virus, you've got thousands of them. Instead of one cell maturing, you got thousands of them. And instead of one cell releasing a few viruses, you've got thousands of them releasing what? Thousands of viruses. And they usually call viruses that go through this acute, because it's not just like you've slowly got this virus coming on you. It's like they come on you all of a sudden, and it's very, very severe all at once. Yeah, so here are examples of viruses. So AIDS, chickenpox, common cold, hand, foot, mouth disease, hepatitis, herpes simplex, and herpes zoster, flu, influenza, Lassa fever, measles, yellow fever, West Nile virus, Marburg fever, mononucleosis, mumps, polio, rabies, rubella, SARS, smallpox, viral encephalitis, viral meningitis, and viral pneumonia. What have we got for a lot of these, and why have you not had more on this list than you could have? What do we do to keep from getting a lot of these? Yeah, is there a lot of debate on vaccines? Yeah, a lot of debate on vaccines. I'm not even going to go into that. But vaccines, if taken, you can eliminate almost everyone off this list for having it, right? So why? How does a vaccine work? Yeah. So they kill the virus, but they inject it into you, and your body don't know that it's dead. Your body thinks it's what? Alive. So your body makes the white cells to fight it off. And then the next time you're exposed to it, you already know he's what? Bad, and you kill him before he has time to what? Take hold. Right. And then that way, if you've been vaccinated for it, you don't have to worry about it because you've been. Does the vaccines work 100% of the time? No. How many of y'all were vaccinated for chickenpox? Just a few? Yeah. So my kids were coming along, my oldest especially, when they first made the chickenpox vaccine. Like my generation, we all had what? We had chickenpox. Like there was people who were like, my kids got the chickenpox, and everybody would go visit them, so all the kids got chickenpox. So then you didn't have to what? Worry about it anymore. Yeah. So why would you want to take a vaccine? Why wouldn't you just get the chickenpox and be done with it? It can. It's very uncomfortable. It only lasts for a few days, maybe a week at the most. So you're like, my kid can suffer through chickenpox. I'm just kidding. But chickenpox also stays in you, that dormant virus, and it can come back. The second round, we don't call it chickenpox, we call it what? That's right. And shingles can be very, very debilitating, very painful. People can get reoccurring shingles over and over and over. If you don't have the chickenpox, does your chances of having shingles go way down? way down, because that's the virus that causes it. It just goes dormant. Do y'all know where the chickenpox goes dormant? In your spinal cord. Does that even sound good? That sounds terrible. And depending on where it's at on your spinal cord is where you get the shingle bumps. So like some people get them, it's very common to get them on their shoulder, your shoulder right here or right here. That's the two most common places, but I've known several people that had them on their what? On their face. So it went dormant up high in their spinal cord. To me, that's getting really close to your what? Yeah, not that it does anything with your brain. It doesn't cause any kind of brain anything, but just the thought of that sounds what? It sounds bad. So I got all my kids vaccinated for chickenpox and all the other vaccines they were supposed to have. But we went to the beach. It's been about, I don't know, 5 years ago. And the second day, my third little girl come out and she's like, Mama, look, I got all these bumps on my belly. And I'm like, first thing I seriously thought was, oh no, this place has got bugs or something, right? Because I'm like, bumps are caused from bug bites. And so I'm like tearing off sheets and I'm looking through everything. And I said, you're probably just allergic maybe to the detergent they used, because I don't know what else. You'll be fine. We're at the beach. Nobody can get sick at the beach. That's against the law. Just go on. We'll be good. So the next day, she's like, I got more. And then the next day, we went home. And so my mom was at my house, and I was like, look at her. She's got bumps on her belly. Tell me what that is. She looked at it like straight off. She's like, that's chicken pox. I'm like, no. She was vaccinated. She can't get chicken pox. She was vaccinated. She's like, I'm telling you, she's got chicken pox. So I went to urgent care at day 4, right, and said, what are these bumps on my kid? And he's like, oh, that's chicken pox. And I'm like, no, she was vaccinated. And he said 1 out of 5 get them anyway. So is the vaccine perfect? It is not. So she was the lucky one who got them. The other siblings called her Pox for like 6 months. They're like, come on, Pox. And she cried every time they said it. So they said it more, right? Yeah. And all the rest of them were fine. So even though she got the vaccine for whatever reason, her body didn't recognize it, didn't form an antibody to it, and she still got it. So you know what the urgent care doctor said? I'm like, okay, so how many days out of school? What have we got to do? He said, go back. He said, go back. He said, everybody's supposed to be vaccinated for it. Don't worry about it. So I tell my kid, I'm like, do not breathe a word, because when it's this huge outbreak of chicken pox, they're going to bring it back to you if you say something. You're going to be Typhoid Mary, right? And so she's like, okay. And then, like, that Wednesday night at church, like, 10 parents came up to me and were like, I heard Lily's got chicken pox. And I'm like, what? So I was at church. I had to tell the truth, right? I'm like, yeah, she got them, but I don't know how all of y'all knew. And so I got my kid. I'm like, what? I told one kid. I swear I just told one kid. Like, that's all it takes. It's all it takes. They're like viruses. It goes like wildfire. Nobody else I knew, though, ended up getting it. But, yeah. So viral infectious diseases, and all of these can be had by a human. Most of these we've had some exposure to. So the flu, how many people have had the flu? Yeah. Common cold. You've had some exposure. Even though you haven't had the current exposure, you still have a little bit of immunity to it. But what makes SARS and West Nile so bad is those viruses have never been here in the United States. So have we ever been exposed to them at all? We haven't. So would we have any immunity to those? We would not. And that's what makes them of such high concern. So the first cases of SARS that came here, and I don't remember the exact country, but it's in the Oriental area. Some people had planned a trip to come to the United States, and they came down with a severe case of SARS, and it's like a respiratory flu-type virus. That's what it does to you. So they had planned this trip, and they had paid their money, so they really wanted to what? To come, right? Do you see their dilemma? They really wanted to come on their trip, so they just got on a plane and came on. So what did that mean to everybody in that airplane that was breathing the same air? If they were United States folks, they had zero immunity to it. So somewhere between flying from over there to over here, they realized these people had this sickness. They were probably pretty sick for them to realize it. They quarantined the whole plane, so everybody that was on that plane could not what? They could not leave. They took them and they put them in a hospital, and they started treating every single one of them because none of us had what? Any immunity to it. There was lots of talk. There was a lot of people who was very angry about that. And they were saying we need to make laws that if you travel when you're sick, that you can be put in jail and you can be fined and you can be this and that. Should you use common sense and judgment? You should, but does people always do that? They do not, and usually they don't. So, yeah, here is a rhinovirus, which is a common cold. This is what it looks like magnified many thousand times. Here's what the measles virus looks like. You can see it has this very unique structure, this glycoprotein outer covering. It's an RNA virus, and inside that, it's an isohedral nucleocapsid, so it's got the sides on it too. And then here's the rabies virus. It's got a really weird shape as well on the outside of it. And here's herpes simplex virus. All of these look different, but they all have, what do all viruses have? Capsid and an inner core, right? This is papillomavirus or genital warts. This is hepatitis virus, so it's a liver cell virus. This is Ebola. It attacks cells that are important to the function of your lymphatic tissues. We know we had that big Ebola scare. They said the guy that had Ebola was on a sidewalk in this city vomiting violently. Can we pull all that virus up and kill it? No. What happened to that virus? Yeah, we didn't see an outbreak of Ebola, though. How come? And Ebola is a weird virus. So even in the countries where it's prevalent, it has times where it's dormant. And they don't know what makes it go dormant, but it'll just quit working. And everybody there will be fine. And then all of a sudden, you'll have an outbreak, and everybody in the whole city will what? Get it and die. And die. It's terrible. Have y'all ever read anything about Ebola? It makes your skin fall off, and like you have convulsions, and pretty much they just have to take the person and like leave them because we have no treatment for it. So is that terrible? Do we want this virus in our country? No. For some reason, it did not make an outbreak. Is it still scary to think that it was here? Very scary, yeah. HIV infects the T helper cells, so basically it destroys your immune system. So usually people who have HIV die from other diseases because they have no immune system. When I worked at the hospital, I know y'all are going to all find this hard to believe because y'all are all super young, but when I was a kid, like first or second grade is when HIV like came out. Like it wasn't here, and then all of a sudden it's here. There was all kinds of scares. I remember I was like in second grade, and our principal called us into the gym and was like, kids, don't be afraid of HIV. You're at no risk of getting it. And all the little kids were like, well, how do you get it? You know, was the principal going to say that to a bunch of second graders? No, he's like, you don't get it from swimming with somebody. Did people like go on a panic scare? They did, because in the beginning we didn't know what. We didn't know how you got it. Lots of people who were homosexual had it, and a lot of people who had blood disorders had it. Why? Thousands of hemophiliacs died. Why? We didn't check your blood. We didn't know it was there. We didn't check your blood for AIDS. So those people that were taking a lot of blood products were what? They were getting it. Right. So we haven't had a virus that has emerged like that in y'all's lifetime, but it was really like a scary thing. And I know people look back at it now and like, you think you could get HIV from swimming in a swimming pool with somebody? And we laugh about that. But at the time, nobody what? Nobody knew, and thousands of people were dying. By the time I got in college, I worked at Huntsville Hospital, and several occasions, I don't know why this stuck out in my head because we had lots of different scenarios, but several occasions we were sent a sample from a patient. The two that I remember were both lung-like sputum. So they were having cold-like symptoms. They could not get over it. They said, there's a bacteria or something growing in this person's lungs. Can y'all please identify it? And I remember one specifically, our techs tried for days and days looking at it under a microscope, having textbooks, pulling it out. What is this? What is this? It's some type of fungus. They eventually could not identify it and sent it to the CDC in Atlanta, and they sent back a report that said it was a fungus that grew in pine trees. And this person had it growing in his what? In his lungs. Why? Because he had AIDS and he had no immune system to fight it off. If we breathed in that fungus from a pine tree, our immune system was like, hey, that is not us, and it would kill it. It would never take hold. Do we have drugs that could kill a pine tree fungus growing in a human's lungs? No, because that's normally not a what? That's not a thing, right? Right, and so most people who have HIV end up dying from these really weird and bizarre infections that normally they would not what? Not die from. The most common one is pneumococcus that's normally found in cats. So it ends up getting in their lungs and what? Just taking off. Yeah. So smallpox, this is what it looks like. Smallpox and polio enters through the gastrointestinal mucosal surfaces, eventually ends up in the central nervous system. Anybody in here know what your central nervous system is? Brain and your spinal cord. So a lot of people survived polio, but it leaves its toll because it affects your brain and spinal cord. It usually leaves you crippled or disfigured, so like you have drawn arms because it affects the nerves that control the muscles in your arms or your hands or your legs. A lot of people died of polio. Lots and lots of people died of polio. All right, so the best defense is prevention. Vaccines stimulate the production of antibodies. It's not as effective against some viruses because of the mutation rate. That's why you got to get a flu shot how often? Every year. That's what they want you to do anyway. Yeah. And then antiviral drugs, and it says question mark. There are a few antiviral drugs on the market, but just very few. Because to kill a virus, you got to get that drug inside your cell, and usually if you get it inside your cell, it also kills what? Your cell. So antiviral drugs are very hard to make. And the first one that comes to mind is Tamiflu. Any of y'all taken Tamiflu? Yeah, when do they say Tamiflu is the most effective? Yes, the very first day, as quickly as you have any kind of symptoms, because it kills the virus outside your cell. So you got to get it when you just have a few of those viruses outside of your cell. And they never say that it prevents the flu. It just what? Shortens your time of having it. Yeah. And there's lots of, I don't know if y'all have seen them, lots of, I don't know, bad side effects to Tamiflu. They say the first time you take it, it works the best, and after that, it goes down. And they're saying there's some people who take it that it gives them some kind of mental, it's not permanent, but while you're taking it, it can affect your thought process. Yeah. Now, we're through with viruses, and we're moving on to who? Bacteria. Yes. So bacteria are cellular. They're made out of cells. They're microscopic, and they're 1 to 10 micrometers long. Do y'all remember that UM? Do y'all remember the metrics lab? Yeah. UM is micrometers. They are prokaryotes. That means they lack a true nucleus. They do have a plasma membrane, though. They do have an outer wall. It's different than the normal plasma membrane, but it's got a layer of peptidoglycan. We're going to talk about that later. Some have a capsule or a slime layer, and depending on which they have, it makes them either harder or easier to kill. Some move by flagella, so they got this big old long whip-like tail that they move by. Some of them have fimbriae, which are short hair-like filaments, and that usually helps them to attach to the area where they're supposed to be. I always say fimbriae are kind of like Velcro, you know, like one side of the Velcro only sticks to the other side of the Velcro, and that's the way these fimbriae work. They do not have a nucleus. We kind of said that right here in 2, right? They do not have a nucleus, but they do have DNA, and it's in a circular chromosome called a nucleoid. And they may have accessory rings of DNA, and we call these plasmids. Tons of super cool research is being done on plasmids, and we're going to talk about that as we go on through here. But plasmids is an accessory ring of DNA. So let me ask you this. What is an accessory? So just like in the regular, what? Yeah! Are accessories 100% you got to have it? No! So I always say this in anatomy. We talk about accessory structures a lot. I'll say, are your pants an accessory? And there'll be some kid in there that goes, yeah, and I'll go, what? If you come out of your house with no pants, you're going to prison. Something's wrong with your brain, right? Like, I mean, some kind of pants of some kind. Yeah, pants are a necessity. They are not an accessory. But what about your watch? If you come out of your house without your watch, you're going to be okay. You're not going to prison. But does your watch have a function? It does. You can do all kinds of stuff with your watch, but it's not detrimental to your livelihood. So that's the way these plasmids are. They have some functions, but they're not, like if they're not there or if they're extras or missing, it's not going to kill the cell altogether. And also bacteria have ribosomes, but they're smaller. So tell me one more time, you already told me once. Ribosomes are the site of protein synthesis. So does bacteria make proteins? They do. They do. Whoops. All right. So here's what a flagella looks like. Let me make sure I didn't do that too quick. Yeah. All right. So this is a bacteria with many flagella, and it has this type of structure because these flagella will go all the way around, 360 degrees. They turn all the way around. It's kind of like a hook that goes inside the plasma membrane, and since it's curved, it don't pull out, but it still allows it to go all the way around. So, reproduction in prokaryotes. It's asexual reproduction, and we call that binary fission. Got to know that for the test. So, bacteria reproduce how? Asexually, and the process is called what? Binary fission. Yes. It takes about 12 to 20 minutes. Is that super quick? It is. I always tell this horribly gross story. So it's picnic time, 4th of July. Everybody loves Aunt Sally's potato salad, right? So Aunt Sally's mixing up her potato salad, but she has to run to the restroom. She forgets to wash her hands when she comes out. And she just drops a couple of E. coli bacteria into that potato salad, just 2. That's not enough to make anybody what? Sick, right? So Sally stirs up her potato salad. She goes, and that potato salad sets out for 1 hour, 2 hours, 3 hours. Now it's noon. She got there about 9:30, 10 o'clock. Now it's noon, 12:30, 1 o'clock. And in the first 12 minutes, they doubled. And the next 12 minutes, they what? Doubled. And the next 12 minutes, they doubled. And the next 12 minutes, they doubled. And so instead of 2 E. coli bacteria in that potato salad, now it's got what? Thousands. Because when you get up to, you know, hundreds doubling, and then 200s doubling, and then those doubling, do you get a lot of bacteria produced in a short time. Now everybody that eats that potato salad gets what? They get sick. That's a terrible, gross story, but is that what happens sometimes? That's what happens. They have a high mutation rate. Their DNA can change, and it has to be able to do that because they reproduce asexually. Why does that matter? If you reproduce asexually, your baby is going to be what compared to you? Genetically the same or genetically different? Same. So if I have a drug that kills you, it's also going to kill every one of your what? That's right. So they have to be able to mutate because they're incurring things in their environment that will kill them. If they couldn't mutate, they would all what? They would all die. Right. So binary fission is as simple as this. You got a cell. It grows about twice as long as it normally does, and it makes a copy of its DNA, and then it starts to pinch apart. And at the end, you've got two cells that are about half as long as that one dividing, and it's got DNA in it, and in about 12 minutes it does it what? Again. Right. Yeah. So genetic recombination. So because they do binary fission, they are all genetically the same. However, times call for them to recombine or get new DNA so that they can survive better. So one way that they combine their DNA is conjugation. It's where two bacteria exchange DNA. And we'll see a picture of this in a minute. They actually have this thing called a conjugation pilus, which is a tube, and they hook it to the tube of the next one, and they transfer DNA. They share it. The second way to do genetic recombination is a transformation. It picks up free pieces of DNA from other prokaryotes. So when these bacteria die, their DNA kind of just breaks apart, and there's little pieces of it floating around, and these bacteria can grab a piece and incorporate it into their own DNA. And then the third way is transduction, where viruses carry a portion of the bacterial DNA from one cell to another. On the test, you got to know the difference between all three of these. People usually get conjugation just fine because that word sounds differently, but a lot of times people get these two what? Switched. Right. So make sure you know transformation is just picking up free pieces of DNA. Transduction, you got to see the word virus in your answer for it to be transduction because it's a virus bringing it in. All right. So dormant conditions. Bacteria sometimes go into what we call a dormant condition, and they produce what we call an endospore. These are very, very hard to kill. So endospores is a dormant bacteria that has grown this outer covering. It says it allows bacteria to survive and to be dispersed. So let me see what she's got here. They survive the harshest of environments with these endospores, and botulism can do this in canned foods. Let me see if she's got one more. Nope. So let me go back to that one. Yeah. Sometimes you will hear of an outbreak of some type of bacterial infection after a building has been moved, especially if the building was really old. A lot of times dozer worker folks will breathe in some of these endospores that have been buried in dirt for many, many years, but since they're moving dirt, it exposes them, and if they breathe in this bacteria, can it cause them to become very sick? It can. Even though that endospore might have been under that building for 200 years, when it's re-exposed and when it gets added to cells, it can what? Grow out of that endospore coat and make people sick. Yeah. So prokaryotes and oxygen requirements. You've got several on this list. The first one we talk about is an obligate anaerobe. So obligate again means what? You told us earlier. Has to. You're obligated to. Does anybody know what the word anaerobe means? I could be using my thing. Does anybody know what anaerobe means? Without oxygen. Oh, that scratchy makes my teeth hurt. Yeah. So oxygen, if you require oxygen, you're an aerobe. If you don't require oxygen, you're an anaerobe. So an obligate anaerobe is unable to grow in the presence of oxygen. A facultative anaerobe is able to grow in the presence or absence of oxygen. Both of these are pretty rare, especially from disease causers. We tested for these a lot when I worked in the hospital lab, but I don't ever remember, maybe once or twice, having an obligate anaerobe or a facultative anaerobe. Most bacteria are aerobe. They require oxygen. The only time I remember having an obligate anaerobe is when we tested a diabetic patient that had a sore on their foot. Yeah, is a sore on a diabetic patient's foot bad? It is. Why? Yeah, and why don't you get enough blood flow with a diabetic? Their blood is full of what? That's right, because you are not controlling it. And foot even more, because that is the very lowest what? Yeah, so your blood's already given off all of its oxygen. By the time it gets down to the foot, it has no oxygen left, especially if it's full of what? That's right. So this person had a bacteria that was growing in that sore, and they ended up having to have their toe what? Right. Because they did not have enough blood flow down into that had oxygen in it, and it was an obligate anaerobe. Most bacteria that affect us require oxygen because most of our body is highly what? Oxygenated. Right. All right, so then we talk about autotrophs. So what does the prefix auto mean? If you got an automatic car, what does it do? That's right. Auto means self. So troph means eater. Autotroph means... That's right. You feed yourself. Plants are autotrophs because they don't take in food from an outside source. They use the sun and photosynthesis, and they make their own what? Yes. So autotrophs, you can have a couple of different kinds. You can have photoautotrophs, which means they use solar energy to obtain energy, and we really call that what? That's right, photosynthesis. So plants are photoautotrophs, but we also have some other organisms that are chemoautotrophs. So what do you think chemo means? Yes, they use inorganic compounds to obtain energy. They don't have to take them in from an outside source. They self-make these chemicals that feeds themselves. Is that a pretty good plan? I got everything I need within me to feed myself. I don't have to look for food from an outside source. It is, but the thing is this only does very little, so they grow very what? Yes, very slowly. And then that gets us to us, heterotrophs. That means we have to take our food in from what? From the outside, from an outside source. We can't just make it. And there's several different kinds of these. So chemoheterotrophs take in organic nutrients from dead or decaying material. Are we chemoheterotrophs? Yeah, yes and no, right? So do you eat plants? Yeah, they're not usually alive when you eat them, right? They're already dead. Yeah, do you eat meat? Hopefully it is also not alive when you eat it, right? Yeah. This right here, no. Hopefully you don't eat things that are already what? Right. But do we see that in nature? Yeah, we do. And I'm going to talk about that just a little bit. So one example of a chemoheterotroph is an aerobic saprotroph. Aerobic means what? With oxygen, right? And saprotroph means it decomposes large organic molecules into smaller ones. Yeah. And it's critical in recycling matter. Let me go back to that. Yeah, so this summer, me and my family actually had the privilege to go to Yellowstone. Any of y'all ever been to Yellowstone? We got back like 2 weeks ago. I'm telling you, you got to go to Yellowstone before you die. Put it on your bucket list. You got to go to Yellowstone. Really, really neat. But I've also went to Arizona, and it was the exact same experience. Out there, they've got a different terrain than we've got, right? Here, it's real wet and it's real hot. And that's where especially fungi love to live. If a tree falls here in the woods, what eats it up? Yeah, that's exactly right. And a tree won't lay in the woods very long here. A couple of years, even if it was a tree this big around, it'll just be like, you ever seen a dead decaying tree and you could just pick it up and just crumble it up and it's like falling apart? termites, bugs, everything, eat it up. Out there, it's totally different. So we were walking through the woods on this trail, and there was just like hundreds of trees down, and they were still intact. Like it was just a tree and a tree and a tree and a tree and a tree. And so when we were in Arizona, I asked this guy, I was like, did a storm come through here? Like, why are there so many trees down? And he looked at me like I was crazy, and he's like, that's just years of trees dying and falling. And I'm like, years? Like, why would they lay here years? And it's because they don't have the same bacteria and fungi that we do that does what? That breaks it down. So they break down a lot slower, so they're there a lot what? A lot longer. Yeah, definitely. So these guys are our helpers. They recycle really quickly. All right, so now we're going to go through a little section about classifying prokaryotes. And we classify them based on their cell wall type. And this is going to help you next week for your lab with the bacteria. So some of them have a thick cell wall, and some of them have a thin cell wall. And next week, no, today's just Monday. Wednesday, you're going to do a gram stain of them. So the thick walls are gram positive. You got to memorize that straight up for your test and for the lab. So thick walls, we call them gram what? Positive, because when you stain them, that thick wall holds on to this purple dye, and they look purple. When you look at the microscope, after you grew up your bacteria, you're going to stain them, and you're going to do a gram stain. And if you look in that microscope and you see purple bacteria, you're going to say, ooh, that bacteria had a what? Thick wall. And I'm going to call that one a gram what? Positive. Yes. If it has a thin wall, we call it a gram negative because it stains pink. So here's the deal. You're going to do a gram stain, and you're going to first put on purple, and then you're going to wash the purple out, and then you're going to put on pink dye. If it's a thick wall, it don't all wash out, so it stays looking who? Purple. If it's a thin wall and you rinse it out, it's all going to what? It's going to rinse out, and then you're going to stain it pink, and it's going to look how? Pink. And that's why we do gram stains, because we know the bacteria that are gram negative, and we know the bacteria that are gram positive. So I worked in Huntsville Hospital micro lab for about 3 years. And we got specimens from people all the time, and we gram stained pretty much every specimen we got. Why? There's no doctor that's going to treat you just on the gram stain, are they? Every day. So you come in, you say, I got the symptoms of a UTI. What is a UTI? Urinary tract infection. I'm going to put a little bit of your urine on a slide, and I'm going to stain it, and I'm going to tell them it's gram negative. E. coli is the most common cause of a UTI, and it's a gram negative. So that doctor, when I tell him, oop, it's a gram negative, he's going to say, oh, that's E. coli. I'm going to treat you with what? Amoxicillin, which kills E. coli. You're going to go down the road today taking a drug that is going to get you better. Instead of waiting till tomorrow when I have it grown up and I go, yes, doctor, that is E. coli, 100% sure. He's going to already have started what? Treating you, and you're going to already start feeling what? Yeah. Now, he'll always say, if you're not feeling better in about 2 days, call us back, because is there a chance that it's another bacteria, not E. coli? There's a chance of it. The likelihood is a little bit low, but he'll say, call me back in 2 days if you're not feeling better. 2 days, the lab has it identified for sure, and he'll change your medicine if that's the case. But treatment is done by the gram stain every day, all day long, multiple times, just by the shape and the positive or negative look of your gram stain. Yeah, so based on shape, you got to know this all day long. Spiral is called Spirelli. Rod-shaped bacteria are called Bacilli, and round-shaped bacteria are called Cocci. On the lab, you'll start it today, but you'll actually do it on Wednesday. You're going to have to look in the microscope and see if you see Spirelli or Bacilli or Cocci. So do you have to know those words? You do. Super easy. So here are examples of both, but I'll go ahead and tell you, this is not what it's going to look like on your microscope, because all of these are electron microscopes. So you got kind of that 3D image. These right here, as far as human diseases, are rare. I have seen there are a couple of human diseases not found in the United States so much, but other countries of Spirillum. Bacilli, very common. Cocci, very common. And especially in chains. So doctors know if you say, I've got a gram-positive cocci in chains, he says, oh, that's pneumonia. I'm going to treat you with this particular. If it's in, they call them diplococci, so sometimes they stick together twos and twos and twos and twos and twos and twos. That's another kind of illness. I can't remember them all. It's been a long time since I worked there. When I worked there, we'd say, oh, they've got this, diplococci, very evident. We could not say that, but we would put that in the computer, and then you could look, and the doctor would prescribe the medicine that you thought he would based on just the shape and if it was gram-positive or gram-negative. All right, so we can also classify prokaryotes based on their metabolism, the presence or absence of endospores, nutritional characteristics as well. So prokaryotes are divided into two domains. You got to know these two. Do y'all know them? Yeah, bacteria was the first one, and we just about at the end of it. And who was the last one? Archaea, and bacteria. So I always say to my classes, Archaea are the extremists. So, have y'all ever watched that show with old Bear Grylls? Yeah, I've only seen that show a few times. Is he an extremist? If you've never seen it, they like drop him out somewhere and he has to live and get back, and he eats elephant poop and all kinds of stuff. Maybe not eats it, but he does all kinds of weird stuff. Is that the normal dude? No! And that, me and you are bacteria. We're the normal people. He is the extremist. He's the Archaea. Archaea live in extreme environments. They can do extreme things that we don't normally see happening. So Archaea have RNA, and it has a different sequence of bases than bacteria. If you looked at Archaea and bacteria on a microscope, they would be similar. Like, I probably couldn't tell them apart. You probably couldn't tell them apart. If you are a scientist who studies Archaea, you could. But they're very similar. But they're made of different substances. Hello. Eukarya are believed to be more closely related to Archaea than to bacteria. So they think like evolutionarily they're somewhere like in between a bacteria and a eukaryote. So types of Archaea. Methanogens. They are found in anaerobic environments. So guys, tell me what anaerobic environment is. Without air. Right. They produce methane. Is that good? It is not. It adds to the greenhouse effect. But they produce it. They live in places like swamps where the water is very still, no oxygen, swampy, methane. Yeah. Halophiles, they're the next group. They require high salt concentrations. So they live where the concentrations of salts are very high, like the Dead Sea. Any of y'all ever been to the Dead Sea? I hadn't either. It's on my bucket list too. One semester I had like 3 students who were like, yeah, I've been there. One guy was from where the Dead Sea is. He was like, yeah, that's in my hometown. And I'm like, wow. It is so salty you don't sink. So like you just float. It is so salty. In my PowerPoint I have a couple of people in it, and you can see them floating. And then thermoacetophiles. They survive best at temperatures above 80 degrees, and they also like it to be acidic. So thermo, hot, and acidic. So they live in places like geysers. And I think she's got some pictures of these as we go. Maybe not. All right. You got to know those, though. You got to know all 3 of those. Methanogens, halophiles, and thermoacetophiles. All right. So types of bacteria. Recent phylogenetic studies indicate that there's 12 groups of bacteria. Here's her pictures. Yeah. So this is from Yellowstone. And this is the weirdest thing. There would be like a big old river like this, and the water in it would just be boiling. So why? There's like steam vents where steam is just coming up out of the ground. Why is it like that? Well, say it again. It is. You're exactly right. So all of Yellowstone is on a volcano. It just hasn't erupted in lots of years. But the magma is right down underneath, and it's got like places where it's coming up closer to the surface, and that's why this is all super hot. I seen one, like there was creeks and rivers everywhere. Everywhere we went, we was crossing a creek or a river, and they were real wide, and it was just as clear, and there was not a single thing growing in it. Why? Yeah, most of them were too hot, or they come from one that was what? Too hot, where it run together. It was just the most odd thing. There was one place where we seen fish, and they were, what is the fish? Trout. And they actually put them in those creeks. So like there was no creeks, and we even read this whole plaque that said, when the very first settlers came there, they were like, "Ooh, this is going to be great fishing. Like, we're going to be able to eat good. We're going to have all the fish we can eat." And there was no what? No fish. No fish. And they inserted those trout, and they do every year because they don't live there by themselves. Yeah. Yeah, is that her last slide? Yeah. So I could talk to you all day about geysers and vents, and I'll hold it down. Yeah, so we're through with the lecture. On your schedule, was we only supposed to cover chapter 20 today? Okay, are y'all telling me the truth? All right, let me upload the text.